Okay, class, where we are now, we want to talk about regulation of gastric secretion. In another way, we want to look at control of the stomach. So, in other words, we're looking at regulation of gastric secretion, but we also really are looking at control of the stomach, because not only does this involve secretion, this also involves the motility of the stomach as it attempts to convert the bolus form, which came from the esophagus, into chyme, which should exit the stomach and go into the small intestine. All right, so the gastric phase, I'm going to say of secretion, or really the gastric phase of digestion would even be a better. I'm going to say gastric phase of secretion, but in parentheses, really gastric phase of digestion, has three phases. One is the cephalic phase. Now that involves the head. The next is the gastric, that involves the stomach, and the next is the intestinal. So to say it another way, before you even, the cephalic phase is this. You go and you're passing by a bakery window and you happen to glance in the window and see something good. You're going to already start having some stomach activity, just the visualization, because that's coming from the brain. Now, then, if you eat it, you'll have some cephalic because you'll get the taste. But then if it gets into the stomach, as it gets into the stomach, then you'll have some increase, some more activity. That's the gastric phase. Then actually, as it leaves the stomach going into the intestine, then the intestine has influence on the stomach, even though the intestine is after the stomach. Some of these are more stimulatory, like the cephalic and gastric, and some are more inhibitory, the intestinal. The logic behind that is the intestine, remember, I said it doesn't want to get stuff too fast. See, look at the size in this picture right here. Here's the stomach, here's the intestine. Look how big the stomach is. So the intestine can't handle all the stomach can do. So it needs to get it in little small amounts along the way. So it kind of slows the stomach back and says, hey, don't get into me too fast. Give me time. All right, so let's walk through. The cephalic phase lasts just a few minutes prior to food entry into the stomach. It occurs even if you don't actually get the food, if you desire the food and not depressed or have a lack of appetite. Okay? Depression and everything else can get involved in this, and you won't even have a cephalic phase if you're too depressed, because you'll be concerned, or too worried. You'll be concerned about other things. I'm going to blow this up a little bit over here, if I can. So, the sight and thought of food, the cortex will do that. Okay? It will then send it to the hypothalamus and medulla oblongata, but the output will be via the vagus nerve. So the key to this is neural. The key to this is neural. Now this is the sympathetic. Now over here, we're going over here. Loss of appetite, depression. See, that case you're using the stress system, which would be the sympathetic. Cerebral cortex also. Lack of stimulatory impulse to parasympathetic center. The parasympathetic center would be over here. So see, depression, worry, things of that can do it. Even overexcitement. So you just won the World Series, the Astros. You're so excited, you're not even hungry. See? Because the cortex has overwritten that hunger area. See, the hunger area is really in the hypothalamus. The nuclei in the hypothalamus really are the hunger area. See, in the hypothalamus, you have a satiety center for being full, and you have a hunger center. The cortex could override that. In this case, the cortex is in congruence with this. It said, okay, ooh, that food looks good. See, the hypothalamus only says you're hungry. Let me kind of break this down to you. The hypothalamus could be the hunger center. It could be satiety. Hypothalamus says you're hungry. It really doesn't say what you're hungry for. The cortex does, because the cortex knows what food you like. That's where you think from. Over here, the cortex, think about it, is where excitement, pleasure, all that stuff is coming from the cortex. And where in the cortex? The limbic system. The limbic system. Okay. All right. So we go further. The gastric phase is when it gets there. Lasts approximately 3 to 4 hours after food enters the stomach. Okay, let's go to the gastric. Stomach distension activates stretch receptors. Vagovagal response, which means you send a signal to the vagus and it sends a signal back from the medulla and then the vagus nerve. Okay, that's part of it. So the cephalic phase was totally neural. But I'm going to go here. Food chemicals enter in. A rising pH activates chemoreceptors, G cells, which are gastrin producing cells, release to the blood. These gastrin cells secrete a hormone. G cells secrete a hormone, gastrin, goes into the blood, but the receptors are here on the stomach cells, primarily on the parietal cells, primarily, but also on other cells too, we'll talk about. So stimuli for this phase is gastric distension, peptides, and low acidity. Okay? Now, the peptides, see what you've done is the peptides are proteins. Doesn't even have to be peptides. Now, here's a definition. A protein has more than 100 amino acids. A peptide has less. So that and low acidity, low hydrochloric acid, will stimulate the G cells to do something. Okay? Because they're going to do hydrochloric acid. Gastric distension activates stretch receptors and initiates both the local responses, the myenteric reflex in the nerves, and the vagovagal, both stimulate acetylcholine release. Now, acetylcholine is from the parasympathetic. So that's a neural aspect. That's the neural aspect. Let's go further. Gastrin. See, now we get into the hormonal. Gastrin is secreted by G cells in the pylorus. So the food has to get to the pyloric region. That's where the G cells gets past the body. So when food gets across the pyloric region, then that food, depending on if it has peptides in it or if it's very basic, low acidity, can stimulate this gastrin secretion. Gastrin, the stimulant are partial digested proteins. Caffeine can do it, and rise in pH, which would be more basic. Okay? Now, partially digested proteins, because see, you got to digest them more. Already did some up here in the body of the stomach. High acidity of less than 2 inhibits gastrin, because if you already have high acidity, you don't need the parietal cells to secrete any more what? Hydrochloric acid. Gastrin stimulates release of enzymes, also histamine from the enterochromaffin cells, but its main targets are the parietal cells in the body of the stomach that secrete HCl. Now, see, that histamine being secreted, that gastrin, if it causes histamine, remember the 3 things we already talked about that stimulate the parietal cell? Histamine on the H2 receptors, gastrin on its receptor on the parietal cell, and acetylcholine. All 3 of those really kick off that parietal cell to deliver hydrochloric acid. Go back and study on that. Okay? But over here, these are inhibitory things. Here, see a pH less than 2. We just said that. Emotional upset can also, because the sympathetic nervous system can override. When protein products enter the stomach, the pH generally rises due to the proteins buffering H+. See, remember from your respiratory that some proteins are basic and some are acidic, some amino acids, shall I say. So if an amino acid is basic, it'll buffer the H+. Most proteins have some acidic amino acids and basic amino acids. The rise in pH stimulates gastrin, which causes hydrochloric acid to spew out thus denaturing the proteins. The more proteins, the more gastrin. As proteins are decomposed, the acidity rises and gastrin is inhibited. In addition to G cells being stimulated chemically, they are also stimulated neurally, the parasympathetic, but we just said that, vagus. And sympathetic turns it off. We already showed over here. The vagus was activated in the cephalic phase. We already said that, just the thought. The phase was activated, cephalic and gastric, due to stomach distension. But again, emotional upset can change everything. Regulation of hydrochloric secretion. We've done it, but this is again doing it in my PowerPoint here. 3 chemicals, acetylcholine, histamine, and gastrin, stimulate this. See, I already did that. All 3 are necessary for maximum hydrochloric acid secretion. Antihistamines block H2, decrease HCl. But what we did, we went back, remember, we went back and talked about the H2 antagonist, which would be these antihistamine, but on H2. See, these don't mess with, now I just want to point, these don't mess with inflammation stuff, because that's H1 receptors on cells. These are specialized for H2. But then we also have the proton pump inhibitors, a newer class of drugs. Okay, we've already talked about that. So we've talked about this here. I'm not going to get into that anymore. That was our picture we had. Here we are, regulation, intestinal phase. Okay, so now let's get back to the story. The intestinal phase, brief stimulatory effect as partially enters the duodenum, followed by inhibitory. Okay. The intestine also has some cells that secrete an intestinal gastrin. Now listen to me, an intestinal gastrin. The intestinal gastrin stimulates the stomach, even though it's produced by the intestine. Okay? But that's the only thing that's going to stimulate. Some actions are excitatory and some are inhibitory. The intestinal gastrin stimulates the stomach to continue. However, this action is brief, and that's the only thing the intestine secretes that favors the stomach to keep working, this intestinal gastrin. Everything else is really inhibitory. Let's go further. The action is brief to the fact that the intestinals fill with chyme containing large amounts of H+, fats, partially digested proteins, and various irritating substances. The irritating component is triggered in the form of an enterogastric reflex. Entero means intestine on gastric. The enterogastric reflex is a trio of reflexes that inhibit the vagal nuclei up here, inhibit local reflexes in the stomach, and activate the sympathetic fibers that cause the pyloric sphincter to tighten down. So, in other words, it's mainly inhibitory. So when it gets in here, it feeds back to try to slow things down. Inhibit the vagal nuclei, so the vagus cuts off. Inhibit local activity in the neurons there, and the pyloric sphincter. The purpose of this is not to fill the duodenum with excess acidity and match the small intestinal prostate. Remember, it's much smaller. Additionally, there's a release of several intestinal hormones called the enterogastrins, secretin, cholecystokinin, and VIP, vasoactive, all are inhibitory. So see, you got a lot of inhibitory. You got these hormones which are being secreted. We're going to talk about those. What makes these be secreted, what I'm saying. And you got this trio. So basically, the only positive thing to say increase is gastrin, intestinal gastrin. Everything else is inhibitory to slow things down. Okay, we're going to close here. My time is running on this one, and start here. Response to stomach filling.